Hi, and welcome to my OCRA A-Level Biology revision with me, Christine. So today's lesson I want to look at oxidative phosphorylation, which is the final stage in aerobic respiration. So let's just remind ourselves about some of the products that were made in the 3 stages leading up to this part. So in glycolysis, we have the coenzyme NAD, which has been reduced, and we have 2 of those. We also had 2 net ATP through that process of glycolysis. The glucose was broken down into 2 pyruvates, and they led on to the link reaction and were converted into the acetyl group. Again, we end up with 2 of our coenzymes NAD being reduced. That acetyl coenzyme A was then fed onto the Krebs cycle, and in the Krebs cycle, because we had 2 acetyl groups being brought in, we end up at the end of the Krebs cycle with 6 reduced NAD coenzymes and 2 reduced FAD coenzymes, and because of substrate level phosphorylation, we get 2 ATP molecules. So the key thing before we move on to oxidative phosphorylation is the fact that this is an oxidation reaction. So our coenzymes NAD and FAD have been reduced because they have picked up hydrogens and they are going to transfer them to the cristae. So let's just remind ourselves about the mitochondrial structure. We have our inner and our outer membrane. And in between that inner and outer membrane, we have got an intermembrane space. Now, that intermembrane space is very narrow to allow for the accumulation of hydrogen ions. So that's important in the fact that if we've got a small space, we're going to end up with this accumulation of hydrogen ions. Now, you can see the outer membrane is impermeable to hydrogen ions, so that keeps the hydrogen ions inside that intermembrane space. What we also have is the cristae, which is the folding of the inner membrane. Now, the folding of the inner membrane increases the surface area, and that means that more proteins, electron transport chain, and ATP synthase can be housed within this inner membrane. So this is really important way in which the structure of the mitochondrion is necessary for its function, and its function is oxidative phosphorylation. So let's look at that now then. So we had our reduced NAD, and our reduced NAD is going to come along and it is going to be oxidized, which means that our protein is going to be reduced. Because it's a oxidation reaction, what we're getting is these electrons are going to be able to move through the electron transport chain through a series of oxidation and reduction reactions. Now, as those electrons are making their way through, they are going to be providing the energy to pump hydrogens against their concentration gradient. So moving the hydrogens from the mitochondrial matrix into that intermembrane space. Now, oxygen is the last electron acceptor in the ETC, and therefore, because oxygen is the last electron acceptor, the oxygen accepts the electrons and hydrogen ions and can form water. So this is how water is formed as a waste product of respiration. Now, what was the point in us pumping those hydrogen ions into the intermembrane space? Well, the reason was for them to move down their electrochemical gradient back into the mitochondrial matrix, and that provides the necessary energy for the ADP to be converted into ATP. So adenosine diphosphate, adding in of a phosphate, phosphorylation, creating adenosine triphosphate, ATP. So let's just go back and check this one last time. As the electrons are going to move through that electron transport chain, they're going to release this energy, the energy for those carrier proteins to pump the hydrogens against their concentration gradient into the intermembrane space. That is going to establish a proton gradient. The electrons can recombine with the hydrogen ions and oxygen to form water because oxygen is the final electron acceptor. Those hydrogen ions that we have created, this concentration gradient, they're going to flow down the electrical chemical gradient from that intermembrane space back into the matrix. And as they flow back into the matrix, they're going to move through ATP synthase. Therefore, the motive force provides the energy needed to join ADP and inorganic phosphate to form adenosine triphosphate, ATP. So this movement of the hydrogen ions down their electrical chemical gradient through that ATP synthase and that motive force is what is known as chemiosmosis. So all of it combined together, the electron transport chain for the hydrogen ions to be accumulated so that they can then move down their electrical chemical gradient, provides the necessary motive force to join ADP with the inorganic phosphate to create ATP. That is why it's called oxidative phosphorylation. Oxidative, a number of oxidative reactions which will add a phosphate onto our ATP at the end. So oxidative phosphorylation occurs at the cristae. I hope you like this video, and if you do, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.